Clustering of antigen receptors leads to activation of intracellular signal molecules. Most of the receptors initiate intracellular signaling by the activation of protein tyrosine kinases, enzymes that affect the activity of other proteins by adding a phosphate group to certain tyrosine residues. The receptors for some growth factors provide the simplest example of this type of receptor. They have cytoplasmic domains that contain an intrinsic tyrosine kinase activity. These enzyme domains are normally inactive, but when brought together by receptor clustering, they are able to activate each other by transphosphorylation. Once activated, these tyrosine kinases can phosphorylate and activate other cytoplasmic signaling molecules. The situation in the antigen receptors is somewhat more complex. They do not themselves have intrinsic tyrosine kinase activity. Instead, the cytoplasmic portions of some of the receptor components bind to intracellular protein tyrosine kinases, which are therefore known as receptor-associated tyrosine kinases. When the receptors cluster, these enzymes are brought together and act on each other and on the receptor cytoplasmic tails to initiate the signaling process. In the case of the antigen receptors, the first tyrosine kinases associated with the receptor are members of the SRC, pronounced SRC, family of tyrosine kinases. The SRC family kinases are common components of signaling pathways concerned with the control of cell division and differentiation in vertebrates and other animals. The prototype family member, SRC, was initially discovered as an oncogene, VSRC, which is responsible for the ability of the Rous sarcoma virus to produce tumors in chickens. This viral gene was subsequently shown to be a modified form of a normal cellular gene called CSRC that the virus had picked up from its host cell at some time in the past. Several other common components of signaling pathways that regulate cell growth and division were also first discovered through their oncogenic action when mutated or removed from their normal controls. And that's clustering of antigen receptors leads to activation of intracellular signal molecules. Have a great day.